So this is Yunhao Wang from Dr. Kim Fai's lab at the Department of Computational Medicine and Bioinformatics. Today, I'm very happy to share one of our research works in the transcriptomics field. We developed a very sensitive approach to allow mapping of RNA M6A modification in mouse early embryos. Advances of biotechnology have greatly improved our understanding of life sciences. Let me paint a picture for you of some techniques that have been selected as annual method by the journal Nature Research since 2007. For example, sequencing techniques from the next generation sequencing, as some prefer to call it, second generation sequencing, to the widely used techniques in the past decade, like the single-cell sequencing, single-cell multi-omics, and the currently trending spatial transcriptomics. We are now also witness the emerging of the third-generation long-read sequencing. And that's not all. We have seen in innovative techniques in diverse areas, from like the protein structure mapping and the prediction to advanced imaging methods, as well as revolutionary shifts in stem cell biology and genome editing. Okay, so these techniques are benefiting our research works in almost every biomedical lab, no matter you're working on wet bench or dry bench. Today, I will take you into the world of epitranscriptomics. This field involves various epigenetic or biomedical modifications of RNA molecule in living cells. In living cells, there are more than 150 epigenetic RNA modifications. Among these, the most common and currently best understood is M6A. Over the past decade, some pioneering studies have discovered its writers, such as MET3 and MET14, its readers, such as YTH domain protein family, and its erasers, such as OCBH5 and FTO. In this world of the RNA biology, M6A plays critical regulatory roles in almost all RNA-related processes, ranging from the gene transcription and translation, RNA splicing, exportation, and degradation. I'll spotlight today on the methodology of M6A. The first and most classical M6A mapping methods, such as MIRIPseq, are antibody-based, which use antibody against M6A to pull down RNA fragments with M6A modification site. Recently, some mutation-based methods have been reported. The principle is to use biological enzyme and chemical molecule to convert M6A into another modification. This new modification will cause mispairing during reverse transcription. Similarly, the second solution is to convert unmodified A to be innocent while leaving M6A unchanged, which will cause the mispairing of A to C during reverse transcription. The third mutation strategy is to fuse the RNA editing enzyme ApoBac1 with M6A reader YTH family gene. In this way, ApoBac1 can specifically edit the adjacent of C of M6A into the U under the guide of M6 reader. Third strategy is using the digestion, which use the nuclears such as MAZF, which specifically cuts unmodified ACA motif, while M6A modified ACA motif untouched. But you might be thinking, we already have the widely used antibody-based method, so which have given us vast knowledge about M6A. So why the need for is new mapping method. The reason is simple. Traditional antibody-based methods have some limitations. They cannot achieve the three major research goals in the field. First, map MCC at a single base resolution. Second, provide a quantitative MCC analysis. Third, reduce RNA or cell amount requirement in the experiment. Both mutation and digestion-based methods allow MCC mapping at a single base resolution. Mutation-based method MCC, SAXIC, and ETAMSIC permit quantitative MCC analysis. Of note, while DARSIC can be used for low RNA and cell numbers, it requires genetic experiments. That is a construction and expression of the fusion genes, limiting its application for the in vivo system, such as developing cells and tissues. Therefore, there is a clear need for a new ultra-sensitive MCC mapping approach that can be used practically. And that's why we plan to develop a method that can map MCC in living systems using a low input RNA or cell samples. Another important reason that drives us to develop a new approach is from the knowledge gap in the field. Over the last decade, we have witnessed the major advance in omics techniques. They have uncovered many omics profiles, specifically for early embryo development, including the transcriptome and epigenomes, such as DNA methylation, protein accessibility, histone modifications, and 3D genome structure. After several years of work, and this year we released our ultra-sensitive MCC mapping method called PicoMiripSeq. This innovative method allows us to map MCC in early mammalian embryos, where the genetic materials are limited. PicoMiripSeq is similar to the traditional antibody-based method. It includes several major steps. First, perform cell lysis and remove genomic DNA and ribosomal RNAs. Second, share RNA into small fragments. Third, use antibody to enrich RNA fragment with MCC modified bases, followed by washes to remove background noise. Then construct a sequencing library. Some of you may ask why we can achieve low input MCC mapping using the antibody-based method, while others cannot in the past few years. The reason is that we optimized many important experimental details during the development of the PicoMiripSeq. First, to reduce the loss of the material, if possible, we will perform as many as steps in the same tube. For example, in the steps of the genomic DNA and the ribosomal RNA depletion and the sequencing library preparation. Another critical factor for reducing material loss is to use a low binding tube. The signal to noise ratio is much higher when using low binding tube than the ordinary tube. Sharing the full-length RNA molecule into small fragments, which is compatible for downstream sequencing, is another challenge. When RNA amount is low, we optimize the sonication cycles for different RNA amounts. Next, marching towards the heart of the antibody-based method, the antibody itself, we found that MCC antibody from the company Medipro greatly outperforms others. After antibody binding, the following wash step is also important to purify the true MCC modified fragment from the large background noise fragment. We found that using the high salt condition works better than the conventional wash buffer. So basically, this is how we develop PicoMeRiPseq. After sequencing library preparation, the next step is sequencing. Next generation sequencing platform such as Illumina NovaSeq used in our study are used to generate the sequencing data, and process into the post-sequencing stage. After quality control and adapter remove of sequencing data, sequencing reads are aligned to reference genome. We further remove sequencing reads from the ribosomal RNA loci, those aligning to multiple genomic loci, and those PCR duplicates from library preparation. Using the IP sample and the corresponding input sample, which is generated by regular RNA-seq without IP step, the M16 modified regions, all M16 peaks are identified by the widely used bioinformatics software Max2 that is also typically used for chipstick data analysis. After peak calling, a series of downstream analysis can be conducted, such as genomic localization of M6A and M16 motif search. We first put PicoMeRiPseq to the test with poly-A selected RNAs from the mouse liver at 3 different amounts: 10 nanogram, 1 nanogram, and 100 picogram. Just as in the existing study, which used much more RNA than us, all 3 RNA amounts in our experiment show a very clear M6A profile around the stop codon of the gene Pura. What's more, we observed high data correlation across the 3 different RNA amounts on the transcriptome-wide scale. After peak calling, we even identify twice as many M6A peaks using the 10 nanogram in comparison to the over 30 nanogram of the previous study. As much as 87% of the peaks from the previous study overlap with ours. Further, in terms of the peak number, there is no big difference between 10 nanogram and 1 nanogram, although there are only around 7,000 M6A peaks in 100 picogram. All right, go ahead. So the 86% of them overlapped with 1 nanogram. In addition, the overlap ratio between 2 replicates of 100 picogram amount is over 80%, speaking to the high reproducibility of PicoMeRiPseq even with picogram scale RNA. Now let's turn our focus to the gold standards for benchmarking M6A detection methods. The first is evident in the metagene profile. Same to the existing knowledge, we see a clear enrichment of M6A around the stop codon. The second gold standard involves the M6A motif, as known as RRACH motif. Across all samples, we identify significant RRACH motifs, regardless of the RNA amounts used. What does this tell us? Simply that PicoMeRiPseq can reliably map out the M6A profile, even when using as little as 100 picogram poly-A selected RNA. We further apply PicoMeRiPseq to the in vitro cultured cells. We observed clear M6A profiles in mouse embryonic stem cells with the cell numbers of 1,110. Meanwhile, consistent with the existing knowledge and studies, the M6A peaks are enriched around stop codon, and RRACH motif is significantly detected for all starting cell numbers. Now that we have seen picomyrips sensitivity and robustness lets the cut-off eyes on its specificity. We first use the spiking control RNAs, including M6A modified positive control and unmodified negative control. Adding its two control RNAs with the same molecule ratio into mouse liver RNA. After picomyrips experiment, we detect 373 reads and 2 reads in positive control and negative control, respectively. After normalizing the length of the two RNA controls, the positive control has more than 500 times capture efficiency than negative control, indicating the super high specificity of picomyrips. But we didn't stop there. We want a more comprehensive evaluation at the transcriptome-wide level. So we use mouse embryonic stem cell with the M6C writer MET3 knocks out, when in theory, M6C abundance will be much lower than the wild-type cells. For efficient data generation and better comparison, we designed 3 conditions. First is 12 nanogram mouse liver RNA. Second is 6 nanogram mouse liver RNA plus 6 nanogram wild-type stem cell RNA. The third one is 6 nanogram mouse liver RNA plus 6 nanogram knockout stem cell RNA. After picomyrips experiment and data analysis, we identify over 7,000 wild-type specific M6 peaks, while less than 2,000 for knockout condition. We further found that M6 signal of the knockout specific M6 peaks is much weaker than wild-type specific ones. This just goes to show you the high specificity of picomyrips when looking at the transcriptome-wide level. After method development and evaluation, the next logical step is to put it to use and see what it can do in a truly important application scenario. Our method is designed for target where genetic materials are limited, so early embryo is a straightforward choice for us to apply it. Instead of using mammalian early embryos, we decided to first test the water with zebrafish as a model for 2 reasons. First, it's much easier and more cost-effective to use zebrafish embryos. Second, a single zebrafish embryo is bigger and has more RNA molecules than a single mouse embryo. As shown in the peak overlap, this heat map, we found that over 90% of M6 peaks identified in all 5 single zebrafish zygote samples overlapped with those identified in bulk zygote. Further, about 80% of M6 modified genes identified in single zygote samples of our experiment are also detected in the previous bulk study. All these single zebrafish embryo samples show very clear enrichment of M6A around the stop codon and the significant RRACH motif. Having succeeded with zebrafish, we take up the challenge with single mouse oocyte at GV and M2 stages, along with single early embryos at zygote, 2-cell, 8-cell, and blastocyst stages. We see clear M6 signals in maternal inherent RNAs from the gene TAT3 as shown on left-hand side, and the zygotically activated genes KLF5 as shown on right-hand side. Further, principal component analysis tells us transcriptome-wide M6A profiles can clearly mark or separate different development stages. As seen in other cell types, M6A mainly localized around the stop codon, and the classical RRACH motif is significantly observed across all development stages. To investigate M6A profiles with more in-depth details, we also conduct another study focusing on biological application. This study was published early this year. Rather than using the single oocyte or embryo, we pool multiple oocytes or embryos together at each stage. This allows us to generate more comprehensive M6A profiles over the developing mouse oocytes and early embryo. Let me summarize the major conclusions of this study. On average, for each stage, there are around 12,000 M6A modified regions or peaks, and 5,000 M6A modified genes. We focus on the regulatory roles of M6A in two important events during the early embryo development. The first one is degradation of maternal RNA, also called decay, where some RNAs derived from maternal oocytes will be degraded during the development. The second one is zygotic genome activation, also called ZGA, where the transcription of the genes from the zygotic genome is activated. We found that half of the decay genes and the ZGA genes are modified by M6A, and half of M6A modified decay genes are also targeted by microRNA. Further, nearly 90% of expressed transcription factors are M6A modified. Finally, retrotransposon-derived RNAs are largely modified by M6A, such as the maternally inherent MTA element and the zygotically activated MERVL element. Its results suggest the potential roles of M6A during this critical development window through regulating probably RNA stability and translation. However, future functional studies are needed to uncover the regulatory mechanism with the assistance with our pigment-heuristic method. In addition to the M6A mapping method I have already introduced and the pigment-heuristic we have developed, let me draw your attention to a new technique. This technique uses a totally different principle from the existing method. This new technique is the direct RNA sequencing developed by ONT company. This is also known as nanopore sequencing or third-generation long-read sequencing, different from the traditional RNA sequencing, which uses cDNA. It will convert RNA to cDNA before sequencing. Nanopore sequencing can sequence RNA molecule directly. This allows us to directly detect modified bases like the M6A during the sequencing process itself. No antibody or biochemical treatment necessary. Some bioinformatics methods have been developed to detect M6A using direct RNA sequencing data. Indeed, direct RNA sequencing holds significant promise for providing quantitative M6A analysis on a full-length RNA scale with single base resolution. However, it's not without its challenges. It includes the requirement of a large RNA amount, issues of the sequencing error and signal noise. And also, there is no available positive control RNA standard for data training and the bioinformatics method training. Despite these challenges, hopefully we will be seeing the big advanced in how direct RNA sequencing contributes to the epitranscriptomics analysis in terms of the enabling analysis at a single molecule level and single base resolution in the full length scale and in the quantitative manner. So the development of our experimental approach, picomeuristic, is carried out by our collaborator, Dr. Yanjiao Li, from the labs of Professor Dell and Clownland at the University of Oslo. Finally, I want to express our thanks for the funding support. Thank you.
Any questions?
So for your Oxford nanopore-based sequencing, so the nanopore sequencing depends on the detection of currency. So if you also include consideration of modification, the different signals from different base may have more overlaps. Will that increase the error rate?
Yes, yes. So that's why some methods they detect the modified bases like MCC. They relied on the sequencing error. If in one place in the A, they find obviously there is a sequencing error. They think okay, probably it's a modified A. This is one solution. Another solution probably the better way is to just detect the modified basis from the raw current signal instead of using the sequencing error. We know in direct RNA sequencing, the sequencing error now at this moment is still pretty high. It's up to 5% to 10%.
Okay, thanks.